Hello everyone, and welcome to today's webcast. My name is Greta Lindsay Peterson, and I am the Senior Statistician and Product Quality Manager at Golden Helix. I am very excited to be presenting on a topic that has obviously a lot of interest in this field: mixed model methods and their utility to genome-wide association studies. We have over 800 people registered for this webcast and have actually broken the record for the most number of registrants for any Golden Helix event. Before we begin, I'd like to point out the questions pane in the GoToMeeting panel, where you can enter any questions that you may have as the presentation goes on, and we'll answer as many of them as we can at the end of the presentation. Before we discuss methods that were designed to account for inbreeding, population structure, and batch effects in genome-wide association studies, we should get a very brief overview of the short history of GWAS approaches. This will be by no means a complete history, but will cover commonly used methods and some of the modern approaches. These methods will then be covered in more detail later in this webcast. Once we have a good background on GWAS methods, we'll delve more deeply into a handful of select methods, including regression with principal component correction, EMMA expedited or EmmaX, multiple loci mixed models or MLMM, and genomic best linear unbiased predictors, GBLUP. Given these four models, we'll outline the major differentiators to help you decide which of these models you might want to use for your study. Next, we'll talk about how these methods were implemented in Golden Helix's SNP and Variation Suite software. We'll run through the implementation in SVS and demonstrate how easy it is to get results and visual comparisons in the form of Venn diagrams. Finally, the top results from the methods will be compared. The main dataset that I have chosen for demonstration purposes is the Sheep HapMap dataset, due to the significant number of samples and large number of breeds included in the project. The phenotype I will be using was simulated, more details on that later. The methods and approaches here are relevant to all complex common disease hypotheses for human, animal, and plant species. They are not as effective for model organisms where a few loci explain a significant portion of the phenotypic variance. Since mixed models can be used for both human and animal data, let's start off with a quick poll to see what you are studying. I'm going to go ahead and open the poll. Please take a second and check the category or categories you are working with. All right, just a few more seconds. It looks like most people have voted. All right. Looks like we've got most of everyone. Let me share the results with you. Okay. Looks like we have a pretty decent split between human and animal species, with quite a few plant folks out there. That's great. I want to make sure that everyone feels that these methods can be applied to their data today. A brief background of GWAS. Naive approaches such as correlation trend tests or linear logistic regression assumed independence of samples, which means no technical replicates, no batch effects. The same array platforms are used for cases and controls and processed at the same lab under the same conditions. Minor allele frequency greater than 0.05 and Hardy-Weinberg equilibrium. In other words, a perfectly designed experiment from start to finish. Well, we all know that's not exactly realistic. It was quickly realized that batch effects such as microarray plate effects, DNA source, saliva, blood, cell lines, population structure for humans, inbreeding particularly for animals, and other sources of cryptic relatedness was an issue that could not be ignored. Not to mention the cost associated with genotyping cases and controls led to sharing of control samples. The problem with sharing controls is you cannot ensure the samples were processed in the same way under the same conditions and so on. A way to adjust for the violations of the assumptions of independence was needed. The goal of having methods that handle non-independence of samples, batch effects, population structure, etc. is to minimize false positives and to obtain better and cleaner results. However, it is also important to use a method that does not overcorrect the data and as a result miss out on interesting findings. As results need to be validated, either by a follow-up study or qPCR, it is not revealing to have hundreds of SNPs to interrogate. Reducing the signal-to-noise ratio has been of major concern and focus from the beginning of microarray experiments. Fortunately, we now have methods to help us do just that. In addition to naive GWAS approaches, methods were developed to handle the special case of family-based study designs, such as the transmission disequilibrium test, TDT, and family-based association test, FBAT. However, it is often the case that pedigrees can be very complex or incomplete, and population studies might not consist of only unrelated individuals. Fortunately, mixed linear models work well in any case. Now, there are some assumptions that mixed linear model approaches make, which we'll discuss here shortly. First, let's define what is meant by a mixed linear model and the common terminology used. A mixed linear model is a regression model that takes into account both fixed factors and random effects. The standard mixed linear model formula is y equals x beta plus z mu plus epsilon, where mu and epsilon are normally distributed and the covariance between them is 0. Now, the variance component of mu is includes a kinship matrix, and which we'll talk about here in a minute. The fixed factors can be factors such as sex, age, or known loci associated with the phenotype. Random effects will include the family or population structure or any major batch effects, and that is incorporated in the kinship matrix, which is an n by n matrix that describes the pairwise relationships between the number of samples. It can be explicitly specified based on pedigree information or generated by computing identity by state, identity by descent, or genomic relationship matrices. The null hypothesis for a mixed linear model for GWAS is typically that the variance component attributable to the relationship between samples is 0. To review, first there were naive GWAS approaches, correlation trend tests, regression analysis. Then PCA and genomic control methods were introduced to correct for population structure, stratification, and batch effects. Within the past few years, numerous papers were published with mixed model methods. In particular, EMMA, EmmaX, and MLMM were all developed within the last 6 years. I should note that the same year that the EmmaX paper, Kong 2010, came out, the P3D method was published in a paper by Zhang. These two methods are similar in that they optimize the mixed linear model implementation by making the assumption that the effect of any one individual SNP on the phenotype is negligible and that several loci are associated with the phenotype or the complex disease hypothesis. GBLUP has also been getting attention recently as an alternative approach to rank loci and or to select loci that are predictive of the phenotype. Navigating the sea of methods published in the past 10 years is a topic for another webcast. Suffice it to say, the literature on this topic is quite extensive. There are a wealth of alternative methods to analyze GWAS data in a mixed model framework, but here in the interest of time, and the methods now currently available in SVS, we limit our discussion to EmmaX, an extension of Emma, and MLMM, and GBLUP, and contrast those methods to regression with PCA correction. Since the announcement of this webcast, we have had other methods brought to our attention. While we are not able to address those methods directly today, we will be following up this webcast with a blog post with references and links to other methods attendees have shared with us. Now let's review the methods selected for this webcast. I like to think of Emma, EmmaX, and MLMM in terms of concentric circles because MLMM is a natural extension of EmmaX, which was built upon the Emma method. The original implementation of MLMM by Segura et al. Was made available in 2012 and was easily modified to work directly from SVS spreadsheets. The MLM code written by the authors also contained a very nice implementation of Emma and EmmaX. We are not going to talk about Emma directly, as while it is used by MLMM and EmmaX, the EMMA method is more of use to analyze genotype data for model organisms, whereas our focus today is on GWAS studies of complex diseases. For those of you attending this webcast that are studying model organisms, there are a few tricks to make EmmaX and MLMM have more accurate variance parameter estimates by accounting for loci that explain a significant proportion of the variance component effects. While these tricks are not going to be discussed in this webcast, please feel free to contact us after the webcast and we will be happy to lead you through how to do it. Of the four methods, regression with PCA correction accounts for the relationship between samples by including a certain number of principal components in the analysis. Of course, this means determining the number of components to use, and while there are a few methods and rules of thumb to select the number of components, it is still a choice that the researcher has to make. EmmaX adjusts for the relationship between samples using a kinship matrix. To expedite the analysis, EmmaX approximates the variance components by computing them once and reusing the estimates for all probes. EmmaX tests a single loci at a time, unless directed otherwise, by explicitly including one or more loci. MLMM also adjusts for the relationship between samples using a kinship matrix. This algorithm methodically chooses the additional loci to include in the model. It is a stepwise EmmaX method. The assumption is that multiple loci are associated with the phenotype. GBLUP adjusts for the relationship between samples using a genomic relationship matrix. This method computes the allele substitution effects to determine which loci are the best genomic unbiased predictors of the phenotype. If we compare the methods by what they have in common and how they differ, regression with PCA can include population structure as fixed effects and reports the p-value as well as other regression results. EmmaX can include population structure as fixed effects, uses Emma for variance component estimates, includes a kinship component as a random effect, and can compute a p-value for the regression models. MLMM adds the ability to automate the adding in of multiple loci as cofactors to an EmmaX model. GBLUP also uses Emma for variance component estimates, computes a genomic relationship matrix to use as a random effect in the regression model. GBLUP adds the output of random effect components for the phenotype and computes allele substitution effects. Note, either REML, restricted maximum likelihood, or ML, maximum likelihood methods can be used for Emma to compute the estimates of the variance components. However, the SVS implementation favors REML. Regression with PCA assumes that you have principal components available. The Patterson, Price, and Reich in 2006 published the widely cited Eigenstratt papers. Google Scholar lists over 2,400 citations for the principal components analysis corrects for stratification and genome-wide association studies paper, and 841 citations for the population structure and eigenanalysis paper. Eigenstratt or Eigenstratt-like methods can compute the principal components for the data, and in general the first few components pick up the majority of the largest sources of structure such as population stratification and systematic batch effects. The key to this method is correctly deciding how many components to correct for. Choosing too few components can result in too many false positives. Choosing too many components can result in suppressing all of the signal along with the noise. There are two ways to perform regression with PCA. One way is to perform regression analysis on principal component corrected data. The other is to use the top principal components as covariates in a regression analysis. The EmmaX method was published in 2010 by Kong et al. This method was a way to make their work in analysis for model organisms computationally feasible for genome-wide association studies, especially those studies with 1,000 samples or more. EmmaX assumes that the phenotype is for a complex disease and that all the SNP loci have a small effect on the phenotype trait by themselves. The optimizations the authors made to Emma was to only compute the variance components once under the null hypothesis instead of under the alternative hypothesis and recomputing the components for every SNP. The MLMM method was published in 2012 by Segura et al. And is a stepwise EmmaX method. Like EmmaX, MLMM assumes that the disease under study is a complex one, where multiple loci are associated with a phenotype. Loci cofactors are selected in a stepwise fashion by choosing the probe with the smallest p-value at each step and adding that cofactor into subsequent forward inclusion steps. Since EmmaX is used, genetic and error variances are computed once for each step, but re-estimated at every step. The GBLUP method for genomic selection and prediction was first published in 2008 by Van Reyden. Since then, GBLUP has been used for plant and animal studies. GBLUP provides the best linear unbiased predictors, aka the random effect components for each sample, and the genomic best linear unbiased predictor estimates of the allele substitution effects by marker. The absolute magnitude of the ASCs can be visualized in a Manhattan plot alongside p-values for other methods, and the pseudo-heritability estimate can be used to compare the GBLUP fit with other methods. GBLUP uses a genomic relationship matrix which computes much faster than IBS, and this matrix can also be used for mixed linear model analysis. Mixed models are good to use on datasets that have some inbreeding or population structure or cryptic relatedness is suspected. You will need to first filter your dataset down to samples and SNPs with good call rate. One rule of thumb is 90% call rate for SNPs and 98% call rate for samples. Depending on your project, you may decide to adjust those call rate requirements. Typically, whole genome sequencing data is meant for rare variant analysis, which violates the minor allele frequency requirement. This requirement exists to make sure that the linear regression models have enough data for a good fit. Otherwise, the results can be unpredictable. Which model to use? Well, regression with PCA is a good approach if you have homogeneous datasets or datasets with simple structure that can be captured by just a few principal components. EmmaX is good when you are studying a complex disease on a structured population and assume that all loci have a small effect on the trait. MLMM is good when you are studying a complex disease on a structured population and assume that several loci have a large effect on the trait and the rest have small effects on the trait. GBLUP is useful to obtain estimated breeding values, rank allele substitution effects to find quantitative trait loci, or to define a genomic relationship matrix in structured populations. Now we'll talk about the implementation of these methods in SVS. The mixed model implementation in SVS is derived from the MixMoGAM Python package available from GitHub. This package was written by one of the co-authors of the MLM paper. Note, while Segura et al. Did not implement GBLUP as part of their package, GBLUP needs variant component estimates too, and it was convenient to use Emma's REML functions for this purpose. While there are free implementations for these methods available for Python or R, SVS provides a user-friendly interface and can run directly from a spreadsheet. Visualization is also quick and easy in SVS, and we have used some tricks to get these methods to run a bit faster. Before we get into the demonstration, first let's go over the dataset chosen for today's webcast. Why sheep? Why not humans or some other dataset? Well, I needed to find an interesting dataset that could be used for demonstration purposes. The International Sheep Genomics Consortium is a multinational collaboration of researchers to create a SNP chip to identify markers associated with various traits, including disease resistance and meat, wool, and milk production factors. They call this a HapMap project because they intended the genotype data to be publicly available in the manner of the Human HapMap project. The data from the ISGC was interesting as it contains a large number of samples from numerous breeds and includes data from the X chromosome. The dataset was available for public use on request from the ISGC, and while there was no phenotypic data available, it was not hard to simulate a quantitative phenotype that could provide an interesting demonstration of all four methods. Other datasets referenced in mixed model papers include the Wellcome Trust dataset, the 1966 Northern Finland birth cohort, Thale Cress, maize, as well as various cattle datasets. While some of these datasets, notably the plant and animal datasets, are expected to have cryptic relatedness, The Welcome Trust and Finland datasets are not supposed to have related samples. However, the Kong 2010 and Segura 2012 papers show that their methods outperform naive GWAS approaches on these datasets. In particular, I used the SNP50 Breed V1 dataset from the Sheep HapMap project. This data was formatted in the Plink PED-map format and was easy to import directly into SVS with the genomic position information contained in the map file applied to the spreadsheet on import. This data came from an Illumina 50K SNP array, and the consortium performed some pre-filtering on the data. After their filters, about 49,000 markers were left, 110 of them which were unmapped. These markers, along with the single marker from chromosome Y, were removed from the analysis. An initial principal component analysis on the data indicated that 2 breeds were considerably different from the others. I removed these to focus on the breeds that were more similar. As gender information was not provided with the dataset, and I wanted to include the X chromosome in analysis, gender was inferred by calculating heterozygosity rates for the X chromosome. Samples with a heterozygosity rate less than 0.02 were considered males. This resulted in identifying 1,611 males and 1,081 females. Before computing the identity by state matrix, markers were filtered down to those with a minor allele frequency greater than 0.05, and the markers were also LD pruned to remove markers in linkage disequilibrium within a window of 50 markers, incrementing by steps of 5. A pair of markers was considered to be an LD if the R-squared value was greater than 0.5, according to the composite haplotype method. This left about 45,000 markers. The X chromosome markers were removed from the principal component before the principal components were computed to avoid picking up gender as one of the first few factors in the principal component analysis. The heat map is an identity by state matrix. You can see identity by state captures the gradient of relatedness and is not like identity by descent where you can make assumptions on what the values mean. The first 2 principal components after filtering and removing the 2 breeds that were considerably different is demonstrated in this plot. This plot demonstrates the fact that each breed is more similar to each other than to the rest of the other breeds, but they are well distributed in this pattern. And the International Sheep Genomics Consortium has a nice PDF file that talks about what some of these factors or axes mean. And in general, there is well distribution between global locations for these components. In order to simulate a phenotype, I first filtered markers down to those within predicted gene regions. Then I randomly selected 25 causal markers and required a heritability value of 0.4. I also assumed a chi-square distribution for the effect sizes, and while that was still too highly predictive of a single loci, I added in an error adjustment from a skewed normal distribution. I didn't want my simulated phenotype to be too good, but I did want it to be within an interesting gene region. To summarize the analysis steps, first you'll want to filter your markers and samples to keep only those with good call rates and minor allele frequencies. In addition, LD pruning can be used to reduce the markers used for identity by state estimation. Then the kinship matrix should be computed on the subset of markers. Principal components should also be computed on the filtered LD pruned dataset with X chromosome removed. Mixed model analysis is next. Principal components can be included as additional fixed factors or not, but the analysis should be on the filtered but not pruned dataset. Results can be visualized in a p-value Manhattan plot as well as in a Venn diagram of the top results. While I know that quite a few attendees are customers, for the sake of those who are not familiar with SVS, I will try to explain how to navigate the software as I go through a sample project. Once you launch SVS, you are first greeted with the SVS Welcome Screen. From this screen, you can start a new project or launch an existing one. You can also see important technical support bulletins and announcements. Before I start my demo project, let me show you how to create a new project. We'll click on Create New Project. And for those of you working with non-human data, make sure to change the default genome map to the appropriate one. If your species is not listed in this list, contact us and we can work with you to make sure it is added for your use. Let's cancel out of this since I already have a demo project prepared for today. Once I click on the project, it opens up into the Project Navigator view. In the Project Navigator window, we see all of the datasets and spreadsheets associated with the datasets. There is also the node change log that indicates how spreadsheets are changed and when, and additionally, whenever an analysis is run, the parameters are printed in this log. A user can enter in notes of their own for each spreadsheet or dataset node down in the User Notes box. Project nodes can be flagged with different colors to indicate where to start or stop, or to provide a reference for sharing your projects with colleagues. When I open up a spreadsheet, first I see that samples are in rows, and I have different data columns and data values in columns. I have a magenta column, which I have set to indicate that this is a dependent column. And I have some principal components in columns that I'm going to use for analysis. And then if I scroll over, I have some genotype columns. As well, in this spreadsheet, I also have the inferred gender columns that I can use that as a cofactor in my analysis. Now this is marker map data, which means that there is genomic position associated with the genotypes. So I can click on this green map button, and I can see that for my genotypes I do have chromosome and position information available. Now a marker map is required for both GBLUP and mixed linear model analysis to, number one, ensure that X chromosomes are adjusted correctly, but also to ensure that the data is placed in the correct genomic context in the results. Now, we have two different dialogues, one for BLUP and GBLUP and one for mixed linear model analysis. While I'm not going to demonstrate BLUP and GBLUP today, I will show you the dialogue. We do have two different options. You can output the BLUP and GBLUP spreadsheets, and you can also optionally output the genomic relationship matrix. Again, this genomic relationship matrix computes relatively fast and can be used for mixed linear model analysis. This relationship or matrix computes much, much faster than IBS, and so if you're short of time, this is definitely the place to start. Missing genotype data in this spreadsheet needs to be encoded properly to compute the relationship matrix and to handle the analysis. The options that we currently have available are to treat all missing data as homozygous major allele. And GBLUP is always in the context of an additive model, so this means all missing data would be set to 0. However, if you have columns that have a significant number of heterozygote genotypes, then you would be penalizing those heterozygote calls by always assuming missing was 0. And so we can also use average value for the missing value. Again, if you have a mixed analysis of both males and females in your analysis, you should correct for gender if you have an X chromosome or gender chromosome available. And so you just click on correct for gender, and you would need to have your sex column in your spreadsheet. And if you click on select column, any categorical or binary column will be available, and you just select the appropriate one to indicate gender. If you have a chromosome other than X that's hemizygous for males, you would indicate that in this box and click OK to run. I'm going to go ahead and cancel out of this and open up the mixed linear model analysis dialog. Again, it's in the Analysis menu. And in this dialog, we do have several options. We have a linear regression that only uses fixed effects, so that you have access to use the linear regression function that's available with this package rather than the one that's available through the numeric regression. What this gets you is it allows you to, the option to correct hemizygous males, and also allows you to impute your missing data. We do have the EmmaX method, and the one option that's available for MLMM is you need to choose the number of forward steps to use. It'll go up to 10 steps, and then it'll start the backwards elimination steps. We'll not run MLMM today. We will run EmmaX, and you can either compute kinship IBS matrix while you're running this, but again, IBS matrices take a long time to compute, and we generally recommend that you run it on LD prune data rather than on the entire data set. And so you can select a pre-computed kinship matrix, and I have that already done. So I'll select Sheet and scroll down and grab my IBS matrix. I am going to use an additive model today. I will impute my missing data as average value. I do have an X chromosome, so I will correct for gender. And in addition to that, I'm going to add the first 8 principal components in my analysis, since this is such a stratified data set. And I will also include gender as a covariate. And now I'm going to go ahead and click Run. This is scanning through the data set. First, to identify which markers are, or which columns are marker mapped, and it's also recoding the genotype data into an additive model. For those of our customers who are listening, in the past for regression you had to recode your data into a genetic model, additive, dominant, or recessive. If you already have your data recoded in a model, you can still run this analysis. It will detect that the data has already been recoded. Once it recodes the data, it then computes the eigenvalues of the kinship matrix, and then the eigenvalues of the entire variance component matrix. This takes just a little bit of time. And again, since EmmaX computes the variance components under the null hypothesis, it just has to compute those once and then reuses them for every SNP. So right now it is progressing through the SNPs and applying those variance components to each model. And this analysis may be affecting the audio, so I will just wait until this is done to provide more information. All right, the analysis is finished, and the output spreadsheet contains the p-value, the negative log 10 of the p-value, and the proportion of variance explained for each of the markers. And if we sort the negative log 10 p-value in descending order, I'm just going to click on the column number header, select Sort Descending. I can see that I have several markers that are genome-wide significant, and we'll look into that here in a little bit. I'm going to go ahead and close this. Since I did not run the analysis for the other methods, let me just provide a little brief orientation into these methods. More information on these can be found in our manual, and we will be happy to provide the link to that documentation. The GBLUP estimates, again, provide the allele substitution effects. Since I have a mixed analysis, I did specify the X chromosome and did the gender adjustment. However, the values are going to be identical for all markers except for those in the X chromosome. So for most of the rows, the values are going to be the same for both males and females. The MLMM method outputs several spreadsheets. The first and a plot, the variance partition plot. This indicates at each step how much of the variance was explained by covariates, and by the phenotype, by the genetic factors, and the error. Also output is step information in covariate phenotype or covariate matrix. This spreadsheet helps you to decide how many steps you want to, or how many cofactors you want to include in your model. There are several different criteria that you can use. For our purposes today, we will use the step 6, which includes 6 cofactors, and that's optimal according to modified Bayesian information criterion and extended BIC. Some of the other additional outputs are also included in the spreadsheet, including pseudoheritability, RSS, log likelihood, and BIC. The p-values from the MLMM method are contained in this spreadsheet, and we do have sets of columns for each step. Before step 1 is equivalent to running EmmaX because you are not including any additional cofactors. Before step 2 would be including 1 cofactor. Before step 3, 2, and so on. So I want to go to before step 7 to include 6 cofactors. And here I have the p-values, the negative log 10 p-values, proportion of variance explained, and Manhattan category is a column that can be used for plotting in a Manhattan plot. The reason why we have to use a Manhattan category column for coloring is to account for the cofactors. So if I sort the negative log 10 p-value in descending order, I can see that my 6 cofactors come up to the top. Again, they were chosen because they were the most significant SNPs at each step. And after accounting for those cofactors, I do have several SNPs that are still significant in a genome-wide significant level of negative log 10 p-value of 6. All right. So ways that we can visualize this data. Let's go back to the EmmaX results, and we'll do a Manhattan plot. So I'm just going to click on the column number header and select Plot Variable in Genome Browser. Now this is again a non-human species, so by default annotation tracks are not going to show up. So I'm going to add in my protein gene predictions to our annotation tracks pane. And this is relatively boring with all being blue, so I'm going to color that by chromosome. So I'm just going to select by variable, select variable chromosome, and now I can see the different colors for the different chromosomes. I'm also going to rename this so that I know that this is the EmmaX p-values. To compare the methods, we can add in the other spreadsheets. So let's add in the results from the MLMM method, and I've got that flagged as green so that I know to use this spreadsheet. And again, I want to use before step 7, negative log 10 p-values. And because of those cofactors, I need to use the Manhattan category for that particular set of columns for coloring. By default, since this is sheep data, the cofactor is going to be put at the end. It's going to get a color that does not stand out, so I'm going to set that to red. And now we can see their cofactors. I'm also going to add in, let's change this name. Oops. Accidentally deleted that. Let me re-add it real quick. I'm going to rename this to MLMM. And did I need to change this color back to red? Okay. Now I'm also going to add in a horizontal line at y equals 6 so that I can more easily identify those markers that are genome-wide significant. And I can duplicate this and drag it up to the top as well. Now let's look at one of these values that is genome-wide significant, not including a cofactor. And because of how I created my simulated my phenotype, it is not unexpected that the significant p-value is within a gene region, because I filtered down to gene regions before simulating my phenotype. And you notice that in the EmmaX model, this marker was not considered genome-wide significant. If I click on it, it has a value just under a genome-wide significance level of 6. However, in the MLMM, including the 6 cofactors, this has considerably increased in significance and is now a value of 8.3. So this marker would have been hidden, but now it is accounted for and brought to light, and it can be looked into further in further analysis. The other analysis methods could be added into this plot, but let's go ahead and move on since we're running a little bit behind. And I want to show you how to create a Venn diagram. In order to create a Venn diagram, we need to have differences between the spreadsheets. Since these spreadsheets were all run based on the same SNPs, they're going to, by default, have the same values or same markers in each spreadsheet. So I'm going to take the top 1,000 markers from each data set. And I've already went ahead and selected sorted absolute value of ASC for males in descending order for GBLUP and taken the top 1,000. Let me show you how to do that with the EmmaX results that we created. So I'm going to take our spreadsheet. Again, it's already sorted in descending order when we were looking at it. Now I'm going to go to Select Row Subset, and First in Items, and select 1,000. And we can see that the number of rows has reduced to just 1,000 active. All right, let me just check real quick. The ones that are marked with blue should be the ones that we use. So we've got 1,000 for EmmaX. I've removed the 6 cofactors for MLMM and selected the top 1,000. We already checked GBLUP, so I think we're good to go. So we're going to go to Tools, Compare Variance Across Several Spreadsheets, Add Spreadsheets. I'm going to select EmmaX, MLMM, then GBLUP, and then Regression with PCA. And I'm going to change the names. This was, I selected EmmaX first, then MLMM, then GBLUP, and then Regression. And that quickly created a Venn diagram that not only has the counts of markers that are shared between each data set, but also has the percentage of those markers. And we can see that only 15% of those markers in the top 1,000 were shared by all of these methods. And MLMM does have the most number, or the highest number of different markers found by that method. Well, this is one way to compare results, but let's pop back to our presentation and we can talk about a couple other ways to compare methods. All right, so again, we already looked at the Venn diagram. This is just for the top 1,000 markers. I also created a scatter plot for the top N markers and incrementally looked at the top 50, top 100, top 150, and so on markers to see how many overlap. And as you can see, EmmaX is most similar to GBLUP, and then after that it's most similar to regression with PCA, but there are still quite a few differences. And all of these methods, as expected, are more similar for the top hits. Another way to compare methods is a QQ plot. The regression with PCA QQ plot actually looks really good, but this plot was created using 100 principal components. And so we had to really use a lot of components to correct the data down to a good QQ plot. EmmaX also looks pretty good, but we have the 6, and it looks pretty similar to regression with PCA. However, we do know there are some differences. MLMM looks even better because it follows the line further past a value of 3, negative log 10 value of 3. But MLMM does automatically exclude the top 6 markers because it's incorporated into each and every model for all of these other data points. So if I take out the top 6 markers that MLMM used as cofactors, I get a plot that's similar to the MLM plot. However, you notice that it starts to deviate from the y equals x line above the line at about 2.6, indicating that this has not been corrected enough. So in this case, with this data set, it does appear that MLMM is the most appropriate method to use. In conclusion, mixed models can be a useful tool when applied appropriately. You'll want to use the method best suited for your data, and hopefully I've provided some information to help you with your decision in that regard. Mixed models are still not a cure-all for bad data. However, they can help when you do have stratification due to batch effects and population. Watch out for a blog post to come out later this week, more on mixed models as well as with some alternative approaches. I would like to thank the author of the Mixed-MoGAN package and Dr. Christopher Seabury, who also encouraged us to implement the GBLUP and provided considerable feedback in our implementation along the way, and John McEwen of ISGC for allowing me to use the Sheep HapMap data for my presentation today. The PowerPoint slides for this presentation will be made available shortly, and here are some of the references that we used for this presentation today. Now, if you haven't already done so, go ahead and enter your questions into the questions pane, and we'll go ahead and take a look at some of the questions and try to answer as many as we can in the next 4 minutes. So one question is, population structure is a random effect. Population structure can be random when you cannot completely account for all of the structure, especially in the plant and animal world. With inbreeding and crossbreeding, it's hard to completely identify the pedigree and the structure, and it's also considered to be a random effect because each family cannot be completely reproduced. And we do get, when we think of population structure, we can think of family relatedness, and that is definitely a random effect. Question about Bayes-B type models. Again, there are different models out there. GBLUP is similar to Bayes-B models. We only implemented a few of these methods, and we will be happy to point you to other papers and other methods out there, including those that talk about Bayes-B. Question about do you think any of these approaches can properly correct for confounding the presence of rare variants? These methods are, there's 2 components here. There's the population structure, the kinship matrix, as well as the regression that happens on that matrix. It is doubtful that a regression model would be able to fit very well when we have rare variants. And anytime you have a significant skew, it's going to be harder for the random effects to properly account for that. I'm always hesitant to say that GWAS approaches can be used for rare variant analysis just because by default the assumptions are violated, but I'm sure that we're going to be seeing a lot more research into that area in the coming years. Why should kinship be a set of small number of markers? We want to eliminate redundancy when we're computing kinship. Number one, that makes it run faster, and number two, it does not provide artifacts where it could make samples look more related than they actually are if you include markers that are in LD. Does SVS have an R API? No, SVS does not have an R API, but any of the R packages that are licensed such that the code can be converted into Python, we can work on converting that code, and we use NumPy and SciPy for a lot of our Python numerical routines, and we can use those to implement a lot of the R methods. And along with that, yes, you can use non-LD prune data set for PCA. Again, we generally recommend using the cleanest, smallest data set for any kind of population stratification and kinship matrix identification, but you can always use a larger data set if you want to, but it is just adds redundancy. All right, let me answer one more question. Is it possible to perform an interaction analysis using mixed models? At this time, adding specific interaction components is not available, but we are looking at new features that we can implement, whether it's adding interaction or adding different methods for where we go next. All right, any of the other questions that we did not get to today, we will try to address those as soon as possible via email. Please feel free to email support@goldenhelix.com if you have any additional questions or would like to share your favorite mixed model analysis method with us so that we can share that with the rest of our audience. Thank you and have a good day.